subdivision class. That all. That was the last thing I wrote.
We did till
We did till this suffix for date, suffix of date or for date.
this one, right?
This.
Now, yeah.
Yeah.
Now we were talking about the species concept in which I told you that basically you must know the mayor concept. What is exactly the mayor concept? I told you that he has coined a term. He has proposed the concept of a species and he told that anything which can interbreed among themselves, it is a species. Right? This we have already
Yes.
done. Now, again, we will see some of the theories over here. What you must know from this point, I would like to tell you. We have monotypic species and we have polytypic species. The species in which a pattern of characters is present, a fixed pattern, like only one pattern is present, like in bacteria. It is defined that you have only not a proper nucleus, no, no liver, no lung, no kidney, only a fixed pattern of characters is present, right? While if you just talk about a cauliflower, if you go to a vegetable market only, you will see chota, bada, light green, dark green, or a cabbage. So that means in the case of plants, means plants are a advanced forms. Or not only plants, the animals also. They are means you can't find any two dogs similar. If you are a pet lover, you won't find any. It is very, very difficult to find two dogs similar, and though they are having same liver, same kidney, everything, but there is so much difference in the morphological characters. So this is known as polytypic species, right? But that is also not much
Yes.
important. You can leave that now. So, but these terms are important. Ecotypes, ECATs. What I mean to say, I will tell you over here. We have understood what is a species. What if the you will find maximum characters similar in the case of species. I and you belong to the same species. We have maximum characters similar. Only zero point 0.1 characters they are different. Then in the case of genus, quite difference. In the case of families, more different. In the case of order, much more difference. So this way, this is the hierarchy present. Now, as a biology student, we must know who is the father of biology, and it is Aristotle. He is the father of biology and zoology, right? Theophrastus. Theophrastus is the father of botany and his three books are very very important. Histora Plantarum तो बहुत common है जो कि नीट में आती है. On that day, I taught you species plantarum. Do you remember species plantarum? Today is Historia plantarum. So Theophrastus
Yes.
wrote Historia plantarum. You can be also asked name which of the following books have been written by Theophrastus. So you must know this point. Then, apart from this, why Theophrastus is important? Because he has also given a model of classification, which is exactly known as artificial classification. I told you that usually artificial classification is not that much accepted because only few characters are proposed. Right. Then, for Apide Candole, you must know he has written theories elementary dale botanic. He has given the vascular system in taxonomy, right? And he was the
Right.
person who has classified thallophyta, bryophyta, pteridophyta, gymnosperms, angiosperms, etc., right? Then we
Right.
come
Right.
to the Bentham and Hooker. Bentham and Hooker, again, what is important? They wrote Genera Plantarum. This book is also very important. The biggest classification was given by Bentham and Hooker. It is very very important. उसके अलावा आपको भले ही कुछ ना पता हो, बस आपको ये पता होना चाहिए कि they have given the natural classification, they have given the biggest classification, and they have written a book known as Genera Plantarum. Right? इसके जो मेरिट्स हैं वो
right.
नीट के प्रोस्पेक्ट से तो उतने इंपॉर्टेंट नहीं है, लेकिन हाँ, इसके मेरिट्स क्या है, बहुत, इट वाज अ वेरी सिंपल क्लासिफिकेशन दे हैव गिवन. दे हैव कंसिडर्ड मैक्सिमम ऑफ द कैरेक्टर्स. कोई भी कैरेक्टर इन्होंने छोड़ा नहीं था, सब कुछ इन्होंने ऑब्जर्व किया हुआ था. राइट. Then Carolus Linnaeus. Carolus Linnaeus, again, he is the father of taxonomy. Already we did that. Then, according to Linnaeus, which two books you must know? आपको इनकी Philosophica Botanica नाम पता होना चाहिए, Systema Nature पता होना चाहिए, Species Plantarum. ये बुक्स के नाम आपको पता होना चाहिए कि लिनानियस ने लिखे थे. Are you getting my point? And Genera Plantarum also.
Yes,
Right?
yes.
Right? तो बस
Yeah.
आपको इतना. You want me to scroll up? You want 2 minutes?
No, sure. You can continue. You can continue.
You can continue, right? Okay.
Yes.
Now, again, only you must know that Eichler, he has given the phylogenetic classification. Only phylogenetic also means very little characters, like, okay, the plants are having flowers or plants are not having flowers. The external characters they have studied. Cryptogams, they don't have any flower. Phanerogams, they have the flower. This is the importance. In the cryptogams, we have three divisions. Can you tell me the divisions of cryptogams?
Yeah, sir, could you repeat?
I am asking, basically, can you tell me what are the three divisions of cryptogams?
Uh, no, no.
No, no. Actually, in cryptogams, we have three variety: Thallophyta, Bryophyta, Pteridophyta.
Ma'am, just a minute.
Yeah.
Ma'am, Neil is facing an issue with his laptop. So could you wait for 5 minutes? He will join from his
Right,
mobile.
sir. I'm just joining from the mobile right now. That's
Give him 5 minutes, he'll join from the mobile.
Okay, okay, no problem. So this Neil can tell to me also. Neil?
yeah. We sir. It becomes kind of difficult to prove because screen में issue है. Screen
Okay,
is
okay. You
glitching.
want me to pause for 2 minutes? Sushmita ma'am?
Yes, ma'am. Yes, ma'am. Wait for 2 minutes. He's joining from his mobile.
हाँ,
Okay.
Yeah, sir. Am I
okay. Yeah. Then you must know that in phanerogams, we have two things. One is the gymnosperms, which have flower-like structures, and the another is angiosperms, which have the real flowers. Right? Now,
right?
this hierarchy you must know: the thallophyta, the bryophyta, the pteridophyta, the gymnosperms, and the angiosperms. Right? Now,
Right.
the aids of taxonomy. What are the aids? Means basically हर्बेरियम बस आपको नॉर्मल पता होना चाहिए कि डैश, ये नीट में ऐसे आता है, डैश इज अ स्टोर हाउस ऑफ कलेक्टेड प्लांट स्पेसीज दैट आर ड्राइड प्रेसेस को क्या कहते हैं. अब उसमें उन्होंने ऑप्शंस में उसको म्यूजियम लिख दिया, बोटानिकल गार्डन लिख दिया, तो आपको पता होना चाहिए कि इसको ये क्या होता है. जो शीट में है वो हर्बेरियम होता है. राइट. देन
Yes.
बोटानिकल गार्डन में क्या होता है? बोटानिकल गार्डन में ओनली आपको ये पता होना चाहिए, जो फेमस बोटानिकल गार्डन है वो इंग्लैंड में है. इंडिया का जो सबसे फेमस बोटानिकल गार्डन है इंडिया का, वो हावड़ा में है. और हावड़ा वाला तो नील, आई थिंक यू माइट हैव गॉन. नो.
No, no, no. I've
नो.
not gone.
ओके. एनबीआरआई, हैव यू गॉन टू एनबीआरआई इन लखनऊ?
No, sir. I've not been to West Bengal.
ओके, ओके. नो, 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 नो. एनबीआरआई, इट इज इन लखनऊ, इंडिया, यूपी.
अच्छा.
नो.
No, no. I've not been there. Not been there.
ओके. तो बेसिकली आपको ये नाम पता होना चाहिए बोटानिकल गार्डन की, और ये कहाँ है ये भी पता होना चाहिए. राइट. म्यूजियम में वी
Oh,
नो दैट बेसिकली द स्पेसिमेंस को हम प्रिजर्व करके रखते हैं. जूलॉजिकल पार्क वो होता है जहां पर हम एनिमल्स को रखते हैं प्रोटेक्टेड एनवायरनमेंट में. व्हाट इज की? की डेफिनेशन कई बार आती है, कैसे हम, कैसे किस ट्रिक से प्लांट्स और एनिमल्स को आइडेंटिफाई करते हैं उसको हम की कहते हैं, राइट? की, एनीथिंग।
Right, right.
एंड वन मोर थिंग, द की which is based on contrasting characters, it is called as couplet. Like you can have a purple flower or you can have a white flower,
Oh, yes.
right? So it is a key. Now for thallophyta आपको ये टिप्स पता होने चाहिए। थैलोफाइटा में एम्ब्रियो एब्सेंट होता है, एम्ब्रियोफाइटा में एम्ब्रियो प्रेजेंट होता है। उसके बाद डिवीजन देखी जाए तो आपको प्लांट्स में दो तरीके की मिलेंगी। एक में क्या होता है, मेल गैमीट्स मूव कर सकते हैं और फीमेल गैमीट्स के पास जाते हैं। दूसरी होती है साइफनोगैमी। ये यहाँ पर इसलिए पढ़ाया जा रहा है कि जब आप एल्गी का चैप्टर पढ़ेंगे देन दे विल नॉट टेल यू व्हाट इज जूडायोगैमी। दे विल एक्सपेक्ट कि आपको जूडायोगैमी का मतलब पता होना चाहिए, right? You can be asked in which of the following we have fertilization by zoodiogamy. Zoodiogamy मतलब basically मेल्स स्विम करके फीमेल के पास पहुंच रहे हैं। किसमें मिलती है, ब्रायोफाइटा और टेरिडोफाइटा, राइट? सिमिलरली किसमें ट्यूब से होती है, बेसिकली जिम्नोस्पर्म्स में और एंजियोस्पर्म्स में। नील आ रहा है, नहीं आ रहा? कैन यू हियर मी?
I think there's a network issue. Neil?
या
Oh, is the network issue? Wait, wait, he'll join again.
Yeah, sir, I'm back.
Yeah. So I was actually telling you that you can be asked that tell the in which of the following the fertilization by siphonogamy takes place. So you can tell angiosperms and gymnosperms. Is that clear?
Yes, teacher, it's clear.
Okay. Shall I scroll down or you want the time to read it?
Teacher, could you give time to write it actually?
Okay. You want me to scroll up? And please
Yes,
don't write anything extra. Only write the things which, because this is a lengthy chapter, it is quite less
yes.
important for the need, right? But still, the points which I told you, they can be asked in the exam. Over
Okay.
here, I didn't ask you anything, but if you want, you can just read it.
I just want to write that first line. Meyer proposed the biological concept of
Yeah, yeah,
species.
yeah.
That's
Yeah, yeah, yeah.
yeah, teacher. Teacher, if possible, could you just underline whatever you think is important for me to write?
Shall I scroll down?
I'm just writing the three types. That's all.
Okay.
Um,
I think your network is poor. Your camera is not working.
Boy, there is some network issue only. That's why I disconnected.
Okay.
Yeah, teacher, done.
Like you can be asked, dash a group of related species which have more characters in common in comparison to species of other genera. So that is genus. Then in the
Okay.
case of order, you can be asked, yeah, this you can be asked, family. Dash has a group related with less number of similarities as compared to genus and species. It has the similarities, but the similarities are less similar to genus and species.
Okay. Okay.
Then this is now the second form, Solanum, Petunia, and Datura. They are three different genres, but they come in one family, Solanaceae.
Yes. Yes, teacher.
Their family is same, right? Oh, you have to
Right.
write, right?
Yeah, yeah. Yeah, teacher, I'm done. I'm done.
In history of taxonomy, only you must know the, first of all, he is the father of taxonomy and botany. This you must know. Then the name of basically the books. Then for AP Kitty Candle also, you must know the name of the book. Only this much.
Okay, teacher.
Then the non-vascular plants which don't have any vascular system, they are Thallophyta and Bryophyta.
Yes, teacher, done.
Okay. Now, for Bentham and Hooker, I told you only you must know the book, Genera Plantarum. Then he has told the biggest and the natural classification. Only this much you must know. Nothing else.
Okay. Ma'am, I'm done. Could you scroll down?
Yeah. Over here also only write the name of the books. Carolus Linnaeus, Philosophy Britannica, Systema Nature, Species Plantarum, Genera Plantarum.
Okay, just 1 second.
Be sure you know the correct spellings of these books, especially Philosophia Botanica. Sometimes they give the wrong spelling and then they ask you to pick the correct option. Then one more line, very important. In Philosophia Botanica, they have given the principles of nomenclature. In Systema Nature, they have given the scientific name of the animals. In Genera Plantarum, they have given the detailed description of the plant kingdom. How they can ask you? Dash book contains the description of the plant kingdom. It's Genera Plantarum. Dash book contains the scientific name of the animals. It's Systema Nature. Right, Neil? Because
Right.
in NEET, you will get the indirect questions only.
Yeah, teacher, I'm done. I'm done writing this. Okay.
Shall I scroll?
Yes, teacher.
Now, again for Eichler only you must know that he has given the phylogenetic system classification. Then in this you must know, I would prefer that if you draw this table in which you write that we have two subkingdoms called as cryptogams and phanerogams, and you can write that over here the plants are without flower and over here the plants are with flower. Then in without flower you have three subdivisions called as thallophyta, bryophyta and teratophyta, and in phanerogams you can have two subdivisions called as gymnosperms and angiosperms.
Okay. Okay. Yeah, sir, I'm done with the table, more or less.
Then these definitions, which I have marked, only for herbarium you must know. Then the name of these botanical gardens.
Okay. One second.
So in the next week, Neil, whenever you are taking a break from the school, from Akash, before Thursday, please message me, or please message to the coordinator. On that day, we will have the test again of any two chapters. Right, Neil?
Okay. Yes, yes.
And you have to also message to the coordinator which chapters test you want to give.
Okay. Okay, sure. Yeah, teacher, can you continue teaching right now?
Okay. So you want me to now teach?
Yes, yes.
Okay. Now your camera is still not working, right?
No, teacher, I think there will be an internet issue for a while now.
Okay. Okay. Now, so basically the biggest phylogenetic classification of the plant was being given by Oswald Tippo.
Okay.
Most acceptable classification, Oswald Tippo. Then what we must know for the need prospect, already I told you, when we talk about embryophyta means which have embryo, right? So then we have two types, one which have vascular plants and one which don't have vascular plants. The ones which don't have the vascular, what are vascular bundles?
As in,
What do you mean by vascular plants?
by vascular plants, those plants. No, no, teacher, I don't.
Do you know xylem and phloem?
Yes, yes, xylem and phloem.
Yeah, basically, so I want to tell you that in the case of bryophyta, we don't have any xylem and phloem. And in case of Tracheophyta, we have xylem and phloem.
Okay.
So this is
Okay.
the difference, right? So you can be asked which of the following is a non-vascular plant and which Bryophyta is a non-vascular plant, and which of the following is a vascular plant in that Tracheophyta can be a vascular plant, right?
Yes. Yes.
Then if I talk about the important thing, yeah, the most important or the most accepted classification was been given by Whitaker, which is the five kingdom classification. He has considered the three main characters like whether how much complex the cell is, what is the complexity of the organism, and which mode of nutrition the organisms are following, right? But that is, the five kingdom classification in which he has, and this is the main classification. We don't have anything else now, right? We have algae,
Right.
fungi, and plantae, animalia, monera, all are present, and no one has questioned on this classification, right? Then monera, protista, mycota, plantae, animalia. Then what else we must know? We must know about the three domains. Three domains means archaebacteria, monera, and normal bacteria. Means, but this six kingdom classification was not being accepted. Why? Because now we were talking about only two things. One are the prokaryotes and the other are the eukaryotes. If I talk about prokaryotes, can you tell me what is the structure of peptido? What is exactly peptidoglycan?
I Not exactly peptidoglycans. They are basically structures which are formed from the various micromolecules like amino acids. Then
Basically, the cell wall of bacteria is made up of peptidoglycan. The best answer is
yes,
this, that the bacteria cell wall is made up of peptidoglycan. Now, what is exactly peptidoglycan, which is made up of glucose, mannose, galactose, 2-amino sugar, N-acetyl glucosamine, and N-acetyl muramic acid, right? Then if I talk about the
yes.
amino acids, we have L-alanine, we have L-form. Do you understand the meaning of D form and L form? You might have studied in chemistry.
Um,
No?
no,
No.
no, we haven't studied that yet.
In chemistry, we have dextrorotatory. Suppose if I talk about this structure. So if it is rotating the mirror to the right side, or if we have the structure and if it is rotating to left side. So left is levorotatory. No problem. In science, in chemistry, you will study. It is a complete chapter. So they want to tell you that mostly all the organic compounds, they can either rotate to the left side or the right side. So on that basis, we have the amino acids as D-alanine or L-alanine, right? So this is the structure of polysaccharide peptidoglycan, in which we have polysaccharides also, we have amino acids also, and we have diaminopimelic acid also. Very, very important. Then teichoic acids, these are the features of only and only gram-positive bacteria. Muramic acid only and only in eubacteria, right?
Right, right.
Anything you want to read, or shall I control, means copy, keep on
No,
teaching?
no, no, no. No, no, you continue teaching. I'll figure it out.
Then basically, like eukaryotes, the cell membrane of prokaryotes, it is made up of lipoprotein, lipids and proteins only, right? Means that over here
Right,
also. But the cell wall is made up of peptidoglycan. If I talk about cell wall, they don't have a proper nucleus. They have a improper nucleus, which is known as the incipient nucleus, right? Then, if I talk about their DNA, you will see they have a false chromosome, which is exactly made up of circular naked DNA or the non-histone proteins. right?
Right, right.
Now, movement, I told you they can be motile also, they can be non-motile also. Ribosomes, they have 70S type. The locomotion, what is the difference between locomotion and movement?
Basically, movement can be brought about in a non-living and living body, but locomotion is brought about by only living beings, at least in themselves.
Basically, in movement, we are changing the place. But in locomotion, if I am moving like this also, it is locomotion. Right? So that is why the bacteria, they keep on doing like this. Right? Then, if I talk
Yes.
about how they keep on doing, they have flagella. Then, if I talk about examples of prokaryotes, we have true bacteria, we have archaebacteria, we have blue-green algae. These are the best examples of prokaryotes. If I have to make a paper of need, I can ask which of the following is not a prokaryote, in which I can just for example add protista, or I can add fungi. Right? So it is
Right.
not a prokaryote. Then, eubacteria history. If I talk about eubacteria history, uh, what you must know? First of all, you must know that Louis Pasteur, he's very important because he has studied about the germs. He came to know that basically the bacteria, they have germs and they can be harmful. Then Robert Koch, he obtained the pure culture of bacteria. First of all, he cultured the bacteria and he discovered that bacteria can be cultured, and then only they were able to study the diseases which are caused by bacteria. Right? Then the name of the smallest bacteria is Haemophilus influenza. And the largest bacteria is Pulopistium fischelsomi. Filamentous is not important, but you must know just as an idea like Mirabilis is the filamentous bacteria. But you can be asked only the smallest and the longest one. Right?
Right, right.
I think, Neil, you must revise whatever I have taught till now. No? You have understood everything?
I have understood most of it. जो समझ में नहीं आया वो I'm taking screenshots now that I'm on the phone, so it becomes easier.
Okay. So basically, if I talk about shape, we can have a spherical shape. And if we have a spherical shape, then we call that bacteria as foci. Similarly, we can have a rod-shaped bacteria. The rod-shaped bacteria are known as bacilli, right? Only this much you must know: streptobacillus, lactobacillus, diplobacillus, bacillus anthracis. They all are rod-shaped. There is spirillum, spiral-shaped bacteria. They are quite important. The example is spirochete and treponeme. Vibrio is
Yes.
comma-shaped bacteria, right?
Yes,
Right?
yes.
Now, now, motility in bacteria. If you ask me what is the motility, definitely the motility is they don't have any legs. They have flagella. So sometimes the flagella can be absent, so they are known as atrichous. Sometimes they have only one flagella, so it is monotrichous.
Yes.
Sometimes they have a flagella present at only one end. Sometimes they can
Like
have a bunch of flagella present on both the ends.
both ends.
So,
Okay.
yeah, sometimes they can have the flagella present on the whole body. Like, see, suppose this is a whole body. We have flagella.
But teacher, what would be the purpose of having flagella on the whole body?
Because it is helping in the locomotion, no? What is the function of flagella? Motility.
Motility, yes.
Movement. They don't have any legs,
Okay.
right? Now,
Yes,
this
yes,
is the structure of flagella that is not that much important, but still I will just tell you it has a basal body. In the basal body, basically that lies within the cell wall. Then we have basically one pair of ring which is L shape and another which is called as P shape, right? So this is the
right.
L shape, this is the P shape. The diagram is not that much important. Only you must know it has a basal body, it has a hook and a filament. These are the three parts of a flagella.
Okay.
Then, if I talk about flagella, it is made up of a protein known as flagellin protein, which is a contractile protein which helps in the contractions. Pili, again pili, it is smaller than flagella, but it is also helping in motility. It is also helping in giving the sex. Those which have pili, they are usually males, and which do not have, they are usually females. Right?
Yes.
Now, if I talk about a structure of a bacteria, yeah, you must know one more point that is they are usually common in gram-negative bacteria.
Yes. Okay.
Then, if I talk about a structure of a bacteria, you will see that the outermost layer of the bacteria, it means outside the cell wall, outside the cell membrane, we have a very slimy layer, chickeny layer, which is called as capsule. The presence of capsule make the bacteria more pathogenic, and this capsule is made up of polysaccharides and polypeptides. Usually when anything attacks, so this capsule protects the bacteria. Then if we talk about the cell membrane, which is also known as the plasma membrane, it is again selectively permeable. It allows only certain things to go inside. But if I talk about the wall is made up of peptidoglycan, right?
Yes. Yes.
So basically the gram-positive bacteria, you will see that in them the lipids, they are present, but they are present in the less quantity.
Okay. Okay. Yes.
Very important point. The differences are written here also that basically it is single layered, it is bilayered. It contains peptidoglycan, which is 20 to 18 nanometer thick. This is 12 nanometer thick only, right?
Right. Right.
Now, murin constant, basal body contains. Basal body point is not that much important, but only you must know that gram-positive bacteria, they have murin. They have more murin or they have more peptidoglycan. The cell wall is single layered and the cell wall is more thick. This is the property which makes a gram-positive bacteria different from a gram-negative bacteria, right?
Okay. Okay.
Any problem till now?
No, teacher. No problem.
Huh?
No problem. No. No problem.
Okay. You think I am fast?
No, teacher. No, teacher. I'm able to. I'm able to catch up.
No, if you think I can be slow also, or if you want, I can show you 5 to 6 questions now.
No. No, teacher. It's okay.
Okay. Now, bacteria, the cell wall, it can be dissolved by a lysozyme enzyme. Please remember which enzyme can dissolve the bacteria cell wall. It is the lysozyme. And when this cell wall is
Okay.
removed, then basically these bacteria, they are surrounded by only cell membrane, right?
Yes.
Now,
Yes. Yes.
basically, we must know that in the cytoplasm, we have a fluid of carbohydrate, lipids, soluble proteins, enzymes, etc. If I talk about the cytoplasmic organelles, what are found inside the bacteria? Mesosomes are very, very common. What is the function of mesosomes? These are exactly the functionally mitochondria-like structures, right? Very
Right.
important.
Right.
Peripheral
Yes.
is something which is present on the outer side. Central is something which is present in the center, right? Then,
Right. Right.
apart from that, the bacteria can perform photosynthesis. They have some pigments which are called as chromatophores.
Okay.
So, the pigments which are responsible for photosynthesis, they are chromatophores. Purple bacteria have bacteriochlorophyll A and B. Green sulfur bacteria have chlorobium chlorophyll or bacterioverdin, right? Storage granules, basically are the glycogen granules which store carbohydrate. Means these are exactly like we have some granules to store the fat, some granules to store the carbohydrates. Similarly, they also have different granules. Glycogen granules for storing the carbohydrate, metachromatic granules for storing the volatile granules, and these granules, they are made up of RNA. If I talk about the chromatin material, it is exactly not proper, means not a proper cell membrane is present or a nuclear membrane is present. True chromosome is also absent. They contain only a single chromosome, right?
Oh,
No
yes.
histone protein. Very, very important. No histone proteins are present. And apart from the main DNA, they have also an extra chromosomal DNA, which is known as plasmid.
Okay.
Now, then if you ask me what is the function of the plasmid, different places the plasmids have different function. Suppose if I talk about the fertility factor. So F positive are those which have the fertility factor, that means they are the males, and the F negative are those which lack that F factor and they are the females. So whenever this plasmid it is attached to the main DNA, it is called as episome or the HFR cell, high frequency cell. If a
Oh,
bacteria, we say that it is becoming resistant to this certain drug, that means it has R plasmid. If I say it is secreting colicin, which is basically antibiotic in nature, it is containing cholicinogenic factor, right? Now, if I talk
right.
about
Right.
the nutrition in bacteria, again, they can be photosynthetic. That means they are using the light energy. And wherever they are using the light energy, you will see the topic is photosynthetic, means they are using the light energy. And usually you will say that in them, the photolysis of water is not taking place because the source of hydrogen is some inorganic compound like H2S, right? H2S is the source. Like usually we say CO2 plus H2S. Over here, the water is not the source. That is why photolysis of water is not taking place. Right. If I talk about lithotrophs, in them lithotrophs, the inorganic sulfur is there, and the example is chlorobium and thiothrax. If I talk about photoorganotrophs, in photoorganic trophs, any organic compound can be there. Means an alcohol can be there, an amino acid, any compound which has to donate its hydrogen group. If I am saying chemosynthetic means it is not using the light, it is using any chemical to make the food, right? And now, what is that chemical? It can be iron, it can be sulfur, it can be carbon bacteria, it can be nitrifying bacteria.
Yes, teacher.
Then we are talking about the methanobacteria, which are only found in the marshy places, and they are converting the methane into carbon dioxide. Any problem?
No, teacher. No problems.
Any questions till now?
No, teacher. No, teacher.
Okay. Now, whenever I talk about the heterotrophs means I cannot make my own food. So if I cannot make them my own food, either I will obtain from the dead and the decaying organic matter. So they are the examples of saprotrophic bacteria. If I talk about obligate means if that is not present, then I can't live. So if I'm saying it is obligate saprotrophic, that means it can only feed on the dead organic matter. If I'm saying it is facultative parasite, that means if it is present, then it is okay. If it is not present, then also it's not a problem. These are the points, right?
Yes, teacher.
Now, for all these cases, you must have an idea of the examples. And if I say symbiotic, the best example it is shown by the green plants, where you can say that the atmospheric nitrogen is converted into the nitrogenous compound. Then if I talk about the staining bacteria, again, we can have simple stains like the Gram staining. It was being discovered by Gram, and in which he has used two stains. One is the crystal volate and the second one is the potassium iodide solution. So first they have to stain the bacteria, then you have to wash the bacteria, and after washing, again you have to restain it with pink color, that is known as saffron. So usually the thing which is decolorizing, that is the gram-negative bacteria. Neil, I think for 5 minutes you must at least take a revision.
Okay, sure, teacher. Sure.
From where you want the revision?
Uh, uh, I think, yeah, this page only, this page, because it has cytoplasmic organelles since it has maximum nodes. Yeah, teacher, could you scroll down?
And please make sure that the next time your camera works.
Yeah, teacher. This is just a one-time thing. Yeah, teacher. Done. Done.
Okay. So Neil, we can continue now from the next class,
Okay. Sure.
right?
Sure.
Yeah.
Sure.
Okay. Thank you.
Teacher. Teacher. Teacher.
Yeah.
How much of the chapter is left now?
Okay. The reproduction part
Okay.
and
Sure.
the diseases part.
Okay.
See, it is not
Okay.
necessary that you do the same chapter. I want you for every week you prepare at least two chapters. Like whichever you
Okay.
think you like. See, if you have prepared from Akash also, suppose. So from my test, you can just check that how much you actually know. And like today, you were thinking that you came to know some new terms, then you can just analyze them, right?
Yes, teacher.
Okay, thank you.
Thank you, teacher.